Hello madam, I'm HEERANYAA Nadaraja and I'm from DBT 4A. Today I would like to perform on the simulation of restriction enzyme digestion. Before entering to this simulation, I would like to give a short intro about the restriction enzyme digestion. What is restriction enzyme? Restriction enzyme are the enzymes that identify specific nucleotide sequence in the double-stranded DNA. They are also very important tools for the biotechnologies. In nature, the prokaryotic cell's function has restriction modification system, cleavage foreign DNA that enter the bacterial cells such as bacteriophages, but not host DNA that has been protected and modified by the methylation. Most restriction enzymes will cleavage the DNA at the specific point within a recognition sequence in a measurable way. Moreover, the agarose gel electrophoresis is the technique um, that used in this specific simulation. When the agarose is melted and then cooled in the aqueous solution, hydrogen bonding causes it from a gel. Under the influence of an electric field, a population of the DNA fragments might generated by the restriction enzyme will move through an agarose gel, whereby negatively charged DNA molecules will be drawn to the anode and positively charged molecules will be drawn to the cathode. Their mobility is almost entirely determined by the size, within the largest molecule having the lowest mobility, and the lowest molecule will have the largest mobility. The pore size will be determined by the concentration of the agarose in the gel, and the DNA fragment of the specific size will migrate at different rates through the gel of various concentrations. Let's start to simulate. In this simulation, we will be performing on a restriction digest. We will use the restriction enzyme to cut the DNA strands, as I said before, to produce the DNA fragments. In order to get to know the size of our plasmid and the enzyme will cut the DNA, we can predict the length of the fragments that will be produced, and it also confirms the length of these fragments and determine the length of the fragment produced from the unknown plasmid construct using the gel electrophoresis, such as the agarose gel electrophoresis. And here we have some short briefing of this. The aim of this experiment is to produce the two DNA fragments. One fragment contains the gene of interest and the other fragment will have the plasmid backbone that can be used to express the protein in the bacterial cells. And here we have the two plasmids which are pKa plasmid and PARA plasmids. Let me introduce the materials and apparatus that we are going to use for this simulation. First, materials. Here we have several reagents that we are going to use for this experiment, such as buffer solution, pKa and R plasmid solution, PARA plasmid solution, restriction enzyme, and distilled water. The buffer solution will be at 2.5 times concentration for the restriction enzyme, digest with the purple label, this plasmid solution with the red label, and this plasmid solution will have been with the green label. Here we have the restriction enzyme such as PAM-HIL and HIN with the black label, and here we have the distilled water with the blue label. And here we have the several micropipette equipments. Here we have P2 pipette, P20 pipette, P200 pipette, and P1000 pipette. Usually we use this P2 pipette in between 0.2 to 2 microliters. P20 will be for 2 to 20 microliters. P200 will be for 20 to 200 microliters, and P1000 for 200 to 1000 microliters. And here we are having the tick box for each micropipette. Empty tubes. Here we have 4 empty tubes with the label of A positive, A negative, K positive and K negative. And then we have other equipment such as microcentrifuge, which can be used at the high speed to collect the liquid at the bottom of the tube for the easy transfer, brush to eject the micropipette tip, floating tube rack to hold the solution tubes on the lab bench and inside the water bath, freezer to freeze and set as negative 20 degrees Celsius, which is used to store the solution for the later use or analysis. Water bath, which used to incubate the samples in water at the constant temperature and has the temperature setting and with the timer. An ice bucket to keep the solution at the cold temperature to prevent the temperature fluctuations and enzymatic degradation. Let's perform on the procedure of this simulation. These are the 9 procedures that we are going to do. First, we have to set the temperature of the water bath, then set the volume of the P20 micropipette, and then draw up the reaction buffer into the micropipette. The pipette the restriction buffer into the K positive tube, pipette the restriction buffer into K negative, A positive and A negative tubes respectively, and then dispense the other solutions into the K positive, K negative, A positive and A negative tubes. Then spin the solution tubes and the solution tubes will be incubate at 37 degrees and then it will be stored in the freezer. Let's start. Number 1, set the temperature of the water bath. Have to set the temperature for 37 degrees Celsius. Yeah. And then set the volume of P20 micropipette. Take out a P20 micropipette. We have to set it for 4 microliters. And then attach into the P20 micropipette tip. Is it already attached? Move the restriction buffer to the empty rack. Take out this buffer. And open it. Draw up about 4 milliliters of this restriction buffer. And then we are going to open the K positive tube. Then we're going to dispense this restriction buffer into the tube. After that, we should eject the tip. Next, pipette the restriction buffer into the K negative, A positive and then A negative tubes. So we are going to do the same thing, the same procedure what we did for the K positive tube for all the rest of the tube, which are A positive, A negative and K minus. Make sure we should change the tip for each materials that we are going to dispense. Now dispense about 4 microliters of the restriction buffer into the K negative tube. Then open the K negative, dispense. Okay. Then you have to eject. Again, attach the tip, close the box, and then dispense We have to close it. Then keep it into a positive tube. Then close it. Have to eject the tip. And lastly, we're going to dispense into the A negative tube. That's all. We have to close all the tubes and then we have to eject the tip into the trash. Next, dispense other solutions into the K positive, K negative, A positive and A negative tubes. The other solutions such as this PKNR plasmid, this PARA plasmid, this restriction enzyme and this distilled water into these 4 tubes. So let's go one by one. We have to use the same micropipette with the same volume, which are 4 microliters. Now let's dispense the, okay, we get it back into the buffer. Let's take one by one. Okay, let's open it. Dispense. Okay, let's start in order. Okay, positive. Let's dispense it, close it, and then eject into the trash. Eject the tip into the trash. Going to eject the tip into the trash and then dispense about 4 microliters of PARA into the A positive and then the A negative. So we get it back and then we let the PARA plasmid. So attach the tip. Into A positive and the A negative. Close it. Now change the volume of the P20 micropipette to 2 microliters. This is 4 microliters, so we have to change it to 2 microliters. Here we go. Save the volume. And then dispense about 2 microliters of the restriction enzyme into the K positive tube. So we get it back and then we let the restriction enzymes into the rack. Open it. Before that we have to attach the tip and then dispense and then pour it into the K positive tube. Again, the same method for the A positive tube. Okay. Then dispense about distilled water, 2 microliters of distilled water into the A negative tube. So we are adding the distilled water here. Open it, touch, open, A minus Q. This time we're going to do the same thing. For the kitchen. Okay, that close. Next, open the microcentrifuge and then place the solution tubes into the microcentrifuge. These are the solution tubes, so we are placing all the 4 into the microcentrifuge. Make sure it should be balanced. Check the balance. Yes, it is balanced. And then set the microcentrifuge to spin for 10 seconds and then remove the solution tubes. Close it. Just for 10 seconds. Start. Okay. Then remove the solution tubes and keep it into the rack. Next, open the water bath. Then insert this. Keep all the 4 tubes here. Then set it for 1 hour. Start. Okay. Next, open the freezer and then place the samples into the freezer. Sorry. So this will be the actual results. And here we can see there are the 4 fragments which are K positive, K negative, A positive and A negative. So these are the length of the fragments, the DNA fragments. So here you can see it's from 400 base pairs to 5500 base pairs. And here you can see the A positive has the lowest base pair. So this is the shortest fragment that may migrate for the longest distance. And K negative and A negative has the longest fragment. Sorry, the K negative only having the longest uh, longest fragment, which may migrate the shortest distance only. So that's all from me. Thank you, madam.